So hello, good morning, good afternoon, or good evening, depending on where you are located. So my name is Matthias. I'm the CEO of bespark Bio, and we are a company specialized in process development for viral vectors. And I've been working in process development for viral vectors over a decade now, especially and mainly in the vaccines area. And today, as part of the LabRoots bioprocessing virtual event, I would like to speak about roadblocks that companies that develop novel medicines commonly encounter when they want to bring their products from preclinical to clinical development or even towards commercialization. I would like to speak about how these roadblocks can be lifted, how the development of these novel medicines can be empowered, and I would like also to give you a short case study, a viral vector vaccine-based case study, and then at the end I will speak a bit about future perspectives. So the increasing prevalence of cancer, genetic diseases, infectious diseases actually increased the need for more cost-effective and more innovative treatments. And one of these treatments, or one of this group for treatment technologies, which is very strong and a focus within the biotech and the pharma landscape currently, are viral vector-based technologies. And when we speak about viral vector technologies, this includes viral vector-based vaccines, for example, vectors like measles, herpes viruses, or adenoviruses, and probably everyone remembers the adenoviral vector-based COVID-19 vaccine from AstraZeneca. And there are also immune oncology viral vectors that are used to specifically target cancer cells and activate the immune system to fight those cells. And there will be kind of vaccinia viruses, vectored technologies, or modified vaccinia ankyras or MVA technologies. Another group of viral vectors that fall into that group bucket would be cell and gene therapy vectors. Again, viral vectors, they're used as a transport and delivery system to bring payloads and antigens into a target cell. And in gene therapy, this is often used is AAV, so adenoviral-associated viruses, and in cell therapy, lentiviral vectors. And then finally, we also have a group which are not per se viral vectors, but they have very common patterns and characteristics, bio-nanoparticles. And here we would speak about VLP, so virus-like particles, exosomes, and other cellular vesicles. So as I mentioned, a lot of innovation and development is happening in biotechs, in small biotech. However, not every product, even if it's very has a very good technology behind it, is not making it into the clinics, or even less of those products are making it to the market. And there can be a variety of problems that those companies face. But there's a specific phase in development, which often is referred to be the valley of death, which is happening during the transition from a preclinical to a clinical state. And there can be reasons like lack of good preclinical data or discovery issues. But there are also issues that are associated with more technical reasons. And those technical reasons often are associated with this CMC part. And for those who are not familiar with CMC, this is an acronym for chemistry, manufacturing, and controls. And this are the crucial activities actually that are required to defining a manufacturing process, to define specifications that must be followed to produce a product safely and consistently. So you have to imagine that a lot of these biotech companies that develop those products, they have a very good understanding on the discovery part, on the science. They have a good understanding of the preclinical and clinical part, but often they lack infrastructure, resources, and know-how on this technical CMC part. So they often don't really know what steps do they need to take in order to develop a process which is capable to produce the product under GMP conditions for clinical testing, but also for commercial licensure. And actually, limitations that often happen then at the end of the day is that these products, they have a prolonged development timeline. They come up with a process which is not very good under control, which is not reliable, which is not robust. And in the worst case scenario, this can either lead to early termination because either the GMP batch fails or the specifications are not met and the clinical trial cannot start, all these problems are manifested later in the development cycle when, for example, the clinical data are good, but then it's realized that the process is not scalable, the cost of goods are too high, so on and so forth. So how can this problem be overcome? So how can we actually bridge this gap? And there would be actually an easy way out, just by saying, okay, you need to spend more time and resources in process development before you go into the clinic. But unfortunately, it's not that easy, because often the companies that develop this kind of products, they have a very strong focus on getting clinical data in order to receive funding, or they're already venture capital funded and they need clinical data to move ahead along quickly. So it's not so easy just to say process development needs to be more focused, or there needs to be more time and resources spent. But another way of thinking is, why not making process development more efficient? Why not making or taking the time and the resources that are already available, make the best out of it, and try to get a robust and then phase-appropriate process out of that time to increase process understanding and the success rate. And once the clinical data are good, they can take this as a basis and further develop the process and let the process grow together with the product to make this a success story. So at the end of the day, to empower scientific breakthroughs, you need to be more efficient in process development. So how can you manage this? So how can you become more efficient? And one way of doing that would be during the selection of the unit operations and the technologies that you implement. So make sure that the technologies, the unit operations, the raw materials you implement in your process, they are state-of-the-art and they support your product. Meaning you always have to think of, okay, what scale do I need to cover the market at the end of the day? And is this technology available at that scale to GMP grade? Or for example, if you have a viral vector which is not sterile filterable, and you need to produce under aseptic validated conditions, is this raw material, is this technology available in a pre-sterilized format, or can it be sterilized? So these are kind of quick fixes, but at the end of the day, you need to make sure that the technologies that you implement are supporting your product on the long run. Then another option that you could look at would be implementing prior knowledge, and especially for companies who have their own process development capabilities or even have platform technologies. They could see and look at already products that have been already transferred into the clinics and look at process steps that they can use as building blocks for new processes, for new products, and try to accelerate process development by leveraging this prior knowledge from previous projects. For companies who don't have this capability, probably this is more difficult, and they probably would need some external support for that. A third option could be implementing data modeling. And there's more and more emphasis to leverage machine learning also for process development. And the use of machine learning techniques can unlock valuable insights for process data. You can identify patterns, correlations, hidden relationships that traditional methods might miss. And you can even be able to develop kind of predictive models that can forecast process behavior. And data modeling and so-called also digital versions of processes, so digital twins, have been used in commercial manufacturing for many years, also in different areas. But for process development, this is rather a new topic, and this can be very powerful to help you to guide to become more effective in process development. The fourth option that you can implement to be more efficient could be quality by design. And quality by design, that's a way of designing quality into processes rather than testing it into processes. And this helps you to create more robust and scalable processes. And if you don't really understand how this works and what it is, don't worry. I will go a bit more into detail into this quality by design. But typically, quality by design is implemented a bit later in the development cycle, prior to filing and getting marketing authorization. However, suggesting to implement quality by design early on could have some added values. And I will speak about the details, the added values, but also the limitations of this in a second. And the final point really is digital transformation. So you could look at ways to implement kind of centralized data hubs for seamless data management and analysis. You could try to use digital transformation to optimize workflows and productivity. So given the time that we have available today, I would really like to focus on quality by design principles and really how we can implement this to make process development more efficient. What really speaks against implementing quality by design early on, but how we can use prior knowledge and data modeling in combination to make quality by design also accessible for early project development phases. So quality by design, that's actually, it sounds complicated, and a lot of companies and people are afraid of implementing it, but at the end of the day, it's not rocket science, and it's really something that can help you to better understand your process. And it's really a development approach that's based on science and the risk management, basically. And all the information that you need to know is covered in the ICH guidelines Q8, Q9, 10, and 11. But quality by design, at the end of the day, ensures that the desired quality of a product is maintained by assessing the variables which might impact the quality. And how does this work? So first of all, you have your product profile. So this would be a target product profile, a TPP. Out of the target product profile, you can extract all the information which is required for quality, which would be your quality target product profile. And this is a spelling error here, so it should be QTPP. So once you have that, you can derive your critical quality attributes, which are all the attributes which define critical quality aspects, which then have an impact on a patient's safety, for example. And just to give you an idea, for a vaccine, for example, a CQA could be either the viral vector titer, or it could be the impurity profile, so concentrations of host cell DNA, host cell protein. So once you have your CQAs defined, you look at your process, your manufacturing process that you have, and perform a risk assessment. So you analyze every individual process step, each handling step performed, and then you think, could this handling step, this process step, this parameter have an impact on my CQA? And then you define, first of all, potential critical process parameters, or potential CPPs, and then you would do some evaluation. Either you can do this outside of the lab, so paper-based, so you would evaluate these parameters, or you have to go into the lab and identify whether your CPPs are affecting your CQAs. And once you have an understanding of what are critical process parameters, you go into the lab and then you define a design space around these critical process parameters, which means you try to understand where is actually the area where your CPPs start to affect your CQAs, and in what range, in what specifications they do not. And by that, you generate a pretty good process understanding, and you really help to understand what's happening in your process and what's affecting quality attributes. And once you have done this, you generate the control strategy, and the control strategy is actually a tool that helps you to control the CPPs within the design space. And then you have a very powerful tool, and this is not a one thing that you do once, because you have to do this once you have implemented it. This is a continual improvement, so that means the more data you generate over time, the more you can update and fine-tune this. And now to make this better understandable, just to give you a concrete example. So let's assume you have a production process where you produce a viral vector in a bioreactor, let's say at 36 degrees Celsius. And with a classical approach, you would set your specification very narrow, so between, let's say, 35.5 and 36.5 degrees. So, and then you're going to make, you produce a batch, and then you have a temperature excursion, and you're out of this specification. And if you have a classical development approach with this out of specification result, you don't have any understanding whether this excursion affects your CQA or not. So whether there's a patient risk or not. So you have to discard the batch. But with this quality by design approach, you would have a design space around the temperature in the bioreactor that would probably span 35 to 38 degrees. And whenever the excursion is within your design space, you know that this excursion does not affect your CQA. So by then you can still use the batch. You may take a hit on the yield, But at the end of the day, the quality is not affected. There's no patient risk, so you can still use your batch. So this is a very powerful tool. But as I said, this often is implemented very late. And there's a reason for that, because this is very time and resource intensive. And when we have a closer look how this is happening. So let's say to the left-hand side, this is, I call this shotgun approach. This is how it was done before this design of experiment and quality by design approaches were implemented. So you just start somewhere with your process development. You do a lot of experiments. It's really unstructured approach. You have really not really a process understanding, and once you have the feeling that your process is kind of under control, you repeat the same process several times, and if you get several times the same result, then you say, okay, that's good enough. Let's use this for production. However, when you implement a quality by design approach, as I just said, you would first define your CQAs, then your CPPs, then you would do some experiments in the lab to kind of set the limits for your design space, and then you would design of experiments or statistical experimental work plan to optimize this design. And this is really a lot of work, and it requires that you understand you have a process already kind of in place. You need to define your CQAs, your CPP. So this can be really time and work consuming. So in order to make use and to leverage the advantages of this approach also for early process development, we need to find a way to use this, or to use the added value of a QBD approach without the laborious work-intensive parts of it. And that's actually what we've tried to do. So we tried to find a way that we can use the added value of a QBD approach without, especially at the beginning of process development, without this huge amount of work. And how can this be done? And our approach here is really to leverage prior knowledge and also prior existing data models, so that you make a kind of an educated guess based on prior knowledge, based on data models that are existing, to reduce data points so that you don't have to do all the experiments in the lab anymore. And also that you can pre-select CPPs based on this prior knowledge, so that you don't have to do unnecessary work in the lab. And I think everyone knows the situation that you do a lot of experiments in the lab, and afterwards you realize, okay, this set of experiments did not increase my process understanding. So I haven't learned anything from that, and you have to go back to square one. And you have wasted a lot of resources, a lot of time. And that's what we try really to avoid. We would like to have a decision support tool implemented that helps you before you go into lab, that helps you really to decide what could be potential CPPs. You focus on those, and you also plan your experiments in a way that you leave out certain data points, and you only do what's necessary, and came up with a very efficient way of developing a process, but also increasing process understanding and process robustness. And next, to show you how this could work, I would like to give you a short case study. And this is a viral vector-based vaccine that was developed over a couple of years. It was produced on Vero cells in a stirred tank bioreactor on microcarrier, and it was developed from preclinical until phase 3 readiness. So we really went through all the hurdles that I just described, and also implemented a control strategy, a QBD approach, a classical one, after phase 1 clinical data came in, so before we went into phase 2 development. And this is the process I'm talking about. So the Vero cells, they're cultured to the left in a static cell culture system. Then they are transferred into a microcarrier bioreactor system where they are infected with the virus, and it's a lytic virus, which means that once the viral vector starts to replicate, the cell substrate is lysed and is depleted, and then the virus is present in the supernatant, and it's harvested, clarified, purified, and then you have basically your drug substance. And the data I'm going to show you and the model I'm going to show you, they're really around this virus production phase. So what have we done? So when we focus on the right side of the slide at the beginning. So we took, this was, as I said, we started implementing this QBD approach after phase 1 data came in. So for phase 1, we kind of developed a phase appropriate, I would say now, kind of quick and dirty process, which was at the edge, I'd say, but good enough for phase 1 development. So we took this data as a baseline. We performed this risk assessment of our process. Based on this risk assessment, we have identified 10 potential critical process parameters, and then we went into the lab, and out of these 10, 3 CPPs remained to be important for this kind of process and product. And these critical process parameters that we have identified have been the pH of the bioreactor, then the temperature, and the MOI, so the multiplicity of infection, which is the ratio of viral vectors to cells. And then we had to perform kind of 10 scoping experiments, and then we went into the design space optimization, which was again 28 experiments. And then we took the data from this approach and generated a data model. So a virtual simulation of our process, a digital twin, and then we looked at that, and this is what you can see on the left side. So you have the purple line, which would be the cell number, which starts to decrease once the viral vector titer increases, which is the green line. And as an example for also another CPP and impurity, we have here the double-stranded DNA, which is the blue line, which also starts to increase once the cells are lysed and are depleted due to the infection. And then we took this approach and looked at the data points, and we have applied our prior knowledge, and we have applied this data model to selectively reduce data points. And then we reduced data points, and we went back again into the modeling approach, and it looked at the data model, at the digital twin, and saw, okay, how does this reduced approach affect the quality of our data model and our digital twin? And this is what I'm going to show you here. So again, on the right side, at the end of the day, we were able to reduce data points based on our prior knowledge, and we reduced up to 30% of these experimental data points. And then we used the remaining data points again to generate a digital twin and compared this, and we realized actually that there is not a difference in the quality of the digital twin. This means we were able to reduce the experimental workload by 30% without affecting the quality of the data model. And this really showed us for the first time that it's possible to get a powerful data model out of a process which is developed actually with less data. And to show you, this is also an anime. So it's a virtual model, but you can play around with the parameters and see how this helps and changes. And this is really a virtual imitation of our process. So really you can use these data models, and we used hybrid data modeling to do this, to show and simulate what's happening when you either change the MOI, which is the bottom lane, or the temperature, which is changed now, or the pH. And you see you have the advanced approach to the left-hand side, and you have the full data set on the right-hand side. And it's pretty much the same. The only difference that you realize is the confidence intervals, which are the dotted lines, they get a bit bigger when you remove the data points. But when we selected the data points to be removed, we took care that this is also based, it's not randomly removing data points, just not only removing data points that the models looked as identical, it's data points that we would have removed based on prior knowledge and the data model that we had available. And again, also that you can see that better because of the limit screen size. This is a zoom in. Again, on the left side, this would be our advanced approach. And to the right side, that's the full data set. And again, you can see there's virtually no difference. And we have the same quality of data model with 30% less data points in it. So of course, I understand there is a limitation to it. So we have kind of retrofitted data we already had. And we also did stay within the same system. So we did stay in the vaccine viral vector system. So the next step, and this is what we're currently working on, is how can we make this accessible to other viral vectors, our viral vector systems? So how can we learn from data models, from prior knowledge from one viral vector system for another to really leverage the full potential of this prior knowledge? And at the end of the day, we have a vision that this could look like this. So we would have a project that is coming in. We would evaluate the technology, the viral vector technology, and would compare this to existing data models from different viral vector types, also to prior knowledge from different viral vector types. And then we would, based on this screening, we would use this kind of a crystal ball decision support tool to define the first kind of set of experiments that are needed to be performed in the laboratory. And again, we would really predefine CPPs, we would predefine the data points that we will need to check and try to reduce this to a minimum. And then after this is executed, you generate a very specific process, manufacturing process for this specific project. And you also generate a very specific data set. And then you use this process and this data set to generate an individual model. And then you use this data model, specific data model, this specific process to further optimize the process. And then it will be, in a very efficient approach, it will be ready for technology transfer to a manufacturer. And if you are able to reduce this by a third, then you would be looking at a time frame for 6 to 9 months for this kind of development approach, compared to a classical traditional approach, which is 9 to 12 months typically, where you don't leverage this prior knowledge in these data models. So now finally, when we really look into the future perspective. So all what we try to do is really leverage prior knowledge and data modeling to be able to make an educated guess, to use this as a decision support tool to support efficient process development design of experiments at the beginning. So really what we try to do is avoid all this risk assessment approach, this definition of CPPs, etc. All you would need to know your CQAs. You would need to know you need to have a ground understanding, a basic understanding of the viral vector technology. But then we would use our prior knowledge and data models that we have to really come up with a very efficient way of doing experiments, planning experiments, and then we would reduce experimental runs or experiments of up to 40%. Of course, the limitations still are, this requires prior knowledge and pre-established data models. Not everyone is having that. And this is where probably companies, especially companies that have no internal capability in the CMC aspect or process development aspect, to get external support. And this can either be just companies that provide support with data modeling and establishing these hybrid models, or this could be companies that support biotechs with process development activities in combination with data models. And also what we're not trying to do is really, we are not trying to replace a classical QPD approach. What we try to do is to leverage the added value of a QPD approach early on in process development. But at the end of the day, if you would like to file a product based on QPD, you would still need to do the whole exercise at the later stage of your development cycle. But the advantage also is that you can already leverage the whole data that you have generated during process development. So the first experiment is already compliant with the QPD approach. Everything that you have done, you can use to follow through your QPD regulatory approval. And with that, I would like to say thank you, and also acknowledge not only the company that I'm working with, bespark Bio, but also another company, Novasign, that we used. They are strategic partners of us who helped us with the data modeling. And I believe Novasign CEO, Mark Dürkop, has as well a very interesting presentation as part of this LabRoots online training. And if you are fancy just to learn more about data modeling and hybrid modeling, I encourage you to also listen to this presentation. So thank you, and have a good day.